All right, so last class, we left off talking about the processivity clamp of DNA polymerase. And so in the previous lecture, we discussed how there's lots of subunits that constitute DNA polymerase 3. And of those subunits that constitute it, for DNA pol 3, there's two subunits that make up a dimer that help actually physically keep the DNA polymerase with the DNA strand. Because again, as I described before, this is a DNA polymerase. It's an enzyme, and usually when an enzyme catalyzes a reaction on its substrate and it produces a product, it usually will release from that product to go back towards a new substrate. But here, for DNA pol 3, it's the beta subunits. For DNA pol delta is again for eukaryotes, and pol 3 is for prokaryotes. For eukaryotes, it's pCNA. It's a trimer that constitutes a part of the DNA polymerase multi-molecular complex that maintains this union with the DNA strand as it's going through and elongating. So up to this point, we've been looking at replication on one strand. We've been looking at DNA polymerase 3. When looking at the top DNA strand here, we've been looking at how there's the primase that creates the RNA primer to help provide the initiating step for DNA polymerase 3. And it creates this strand of DNA that leads, that's elongating from the 5' to 3' towards the replication fork. But this is happening on both strands. This is happening on this bottom strand as well as on the top strand. What's important to note is that the helicase, as indicated by this triangular prism on the DNA strand, helicase is driving further into the DNA strand and opening up, extending the replication fork and creating more and more space for DNA polymerase to continue elongating and to continue replicating and creating a daughter strand. But what's also important to note is that there's also, and we call that strand the leading strand. The leading strand is going into and towards the replication fork as helicase is moving forward and opening and melting the DNA further. Now, this is also happening on the top strand as well. But what's important to note is, as we described before, DNA polymerase 3, it will elongate in the 5' to 3' direction. So what's going to end up happening is primase will lay down an RNA primer and it'll elongate. And there's only going to be so much of the template strand available on that top strand to be able to elongate because again, it wants to go from 5' to 3'. What's going to end up happening is as helicase is opening up on this bottom DNA strand, as it's opening up further and creating moving the replication fork further to the right, there's going to be more space here now for primase to lay down a new RNA primer and DNA polymerase 3 to now create a new strand. But these are going to be happening in small discontinuous segments on this top strand. And so we call that the lagging strand. And the lagging strand is typically characterized by these small discontinuous fragments, whereas the leading strand is one single continuous new DNA strand that's being laid down. And so to look at it from another angle, again, these replication forks it's not just a one-sided thing, there's two sides to it. And so as I described before, at the replication fork, helicase is melting the DNA and pulling it open further. The leading strand is elongating towards the replication fork. So helicase, if you can imagine, you can imagine like in football, you have a blocker that's kind of like leading away and creating a space for the ball carrier to go through. Helicase is the blocker, basically creating space, melting the DNA strand, and DNA polymerase 3 is going through and replicating continuously. Now, on the lagging strand up top here, what's going to end up happening is, again, leading strand is going towards the replication fork, and it's always in the 5' to 3' direction. The lagging strand goes away from the replication fork. But what's important to note is here, we're only looking at one side of this bubble. there's going to be another side of the bubble. So now what's going to end up happening is there's another side where helicase is over here opening up the DNA. And so it's going to, what's going to end up happening is while this top strand is the lagging strand over here, there's going to be another replication fork over here where this strand is going to be continuously being elongated towards that replication fork. And it just depends on what fork you're looking at here. So in this fork, yes, Ahava, do you have a question? That's a good question. So when we're looking at this fork here, so when we're looking at this fork of the bubble, this fork, the top is the lagging strand and the bottom is the leading strand. When we look at the other fork on the other side of the bubble, the top will be the leading strand because it's going towards into that replication fork. And the bottom will be the lagging strand because it's going away from that replication fork on the other side of the bubble. And so what's going to end up happening is as helicase is opening up and melting the DNA more on this replication fork, leading strand again will just continuously go in and elongate the new DNA strand. But what's going to end up happening is each time as there's more template strand available for the lagging strand, an RNA primer will come in and help provide the initiation step for DNA polymerase 3, but then it's going to run into the previous fragment. So, for example, let's say these two strands have already been made. As it opens up further, now there's space for an RNA primer to come in, and it's going to elongate and then abut against this fragment here and stop. And so these are going to be, the lagging strand is characterized by small fragments of new DNA that's been elongated. These are called the Okazaki fragments. As you can imagine, it was discovered or characterized by a scientist named Okazaki. But it's defined by these small little fragments that constitute the lagging strand because of, again, it's a space limited problem where as the helicase is melting the DNA, there's only so much space to grow from the 5' to 3' direction. And you have these little fragments that constitute the lagging strand. So as you can imagine, in these, as we're creating this new complementary DNA from the template DNA, especially with these Okazaki fragments, there's going to be lots of RNA primers and lots of gaps that are present on these new DNA strains that we need to address. And a polymerase that helps to address that issue is DNA polymerase 1, as it's referred to among prokaryotes, or alpha in eukaryotes. So it does some of the similar work that DNA Pol 3 does. It will polymerize or promote polymerization in the 5' to 3' direction. It will have a proofreading exonuclease activity in the 3' to 5' direction. But it can also serve as an exonuclease in the 5' to 3' direction. And this is really important because this 5' to 3' exonuclease activity helps to remove those RNA primers. And so what's really fascinating is that it can combine what DNA polymerase 3 does with its exonuclease activity. So you can kind of imagine that it's both a plow or a bulldozer getting rid of the RNA primers while laying down the new DNA that complements that area where the RNA primer was. And so even though it has more functions than DNA polymerase 3, it acts slower than DNA polymerase 3, and so it's less efficient. And so typically when you're having, there's a reason why DNA polymerase 1 doesn't do everything. Yes? So is it DNA polymerase 3 or Pol delta that does the initial segmenting, and then Pol 1 will roll over that in the 5' to 3' direction and destroy the RNA? Yep, it'll remove the RNA primer and lay down the corresponding DNA for that location on the genome. So 3 then 1. Yes. Yeah. I thought you were saying 3' to 5. I thought you were saying 3' to 1'. I was like, no. Yeah, that's right. So to try and illustrate and kind of put the pieces together here, you have, you can think of, we're looking right now at a lagging strand where DNA polymerase 3 has its RNA primer here that will help initiate the elongation. And what's going to end up happening as I described before, helicase will open up the replication fork further, melting the DNA, creating more space for more elongation upstream from it in a 5' to 3' direction. Now, this is DNA being synthesized that abuts up to the RNA primer, so they will not be ligated together. And so there's a space there. That space is important. So take note of that. So what's going to end up happening is that DNA polymerase 1, or alpha now, it's as we described, it's an exonuclease, but it's not making a cut. This is kind of like a docking site or a break in the chain where it can serve as an exonuclease to come in and in the 5' to 3' direction, both remove the RNA primer and lay down and synthesize DNA for that spot. And there's going to be, even though it's not depicted here, there's still going to be a gap between what DNA polymerase 1 just created and what was already there. And so we're going to talk about what helps fill in that gap. Are there any questions so far? All right. So as I just described, DNA polymerase 1 or polymerase alpha will remove the RNA primer and synthesize the DNA that is meant to occupy that spot on the DNA. But again, as is depicted here, there's going to be a gap between where that RNA primer was and the subsequent DNA upstream from it. And so that gap is filled by DNA ligase. It promotes ligation, and it creates the phosphodiester bonds on the phosphate backbone for DNA. And so to try and help put these pieces all together using the bottom half of this figure, you're going to have helicase going into the replication fork, opening it further. This leading strand with DNA polymerase 3, as indicated by this teal ball, will go in continuously, creating a new daughter strand of DNA off of the template strand, going further, going into the replication fork. On this top lagging strand, you're going to get these Okazaki fragments where, as space is created, as the replication fork opens further, primase will create a new RNA primer here. DNA polymerase 3 will use that as an initiation cue to promote elongation and create the daughter DNA strand, and it'll abut against the RNA primer of what's already been created. DNA polymerase 1 will come in, it'll remove that RNA primer, synthesize new DNA that's meant to occupy that spot, and DNA ligase will join all those pieces together to create a full daughter strain of DNA that has no gaps in it. Yes.
Just checking. So when the leading strand meets the lagging strand, will DNA ligase also help get rid of some secretion?
Yeah, exactly. Yeah. Yeah. Yeah.
Does DNA ligase
it's just bringing the segments together. It's creating, when we look at the chemical structure of DNA, the way in which subsequent base pairs on one side of the DNA, if you look at a single strand, the way in which they're bound together is across the phosphate groups to the sugars with these phosphodiester bonds. And so those are typically the easy sites where things are clipped to either remove them or, in this case, combine them together. And that's what DNA ligase is doing. And that's also the site at which it's acting. Brandon.
Strand until it reaches the next part. And then DNA one comes in and replaces the primary DNA. And then ligase.
Yep, That's exactly right.
And just checking this diagram up here, there's a DNA polymerase 1 here on the leading strand. Does that just act once then, just for the very first prime on the leading strand?
So that what that's doing is, and I think this goes back to a question I was describing before. Each bubble will have 2 replication forks, and so looking at the top half of this figure, the bottom strand, it going towards that fork, it's the leading strand. If our reference point is the fork on the left side of the bubble, it's actually the lagging strand, and so they're going to be Okazaki fragments on to the left, and ligase DNA polymerase 1 will replace the primer there, and DNA ligase will then fuse them, anneal them together. Does that help me answer the question? Great. All right, we'll keep moving. So again, putting all of this together, we've done a couple times now, but repetition helps. We have our origins of replication, which are the sites of replication beginning. The initiator proteins will bind to the replication origins, these sites of where replication begins. These initiator proteins for prokaryotes are DNA A. For eukaryotes, it's the ORT complex. And because particularly for eukaryotes, our initiator proteins cannot melt the DNA, it's important to have these helicases. So helicases will separate the strands of our DNA, and for prokaryotes, that's DNA B, and for eukaryotes, that's MCM 2 through 7. And so now that the DNA has been melted and separated, and we have these single-stranded binding proteins that help stabilize it in a single-stranded melted conformation, gyrases there's going to be supercoiling that occurs as a result of that, and the gyrases, type 2 topoisomerase particularly, will be helping to relieve that coiling. And type 1 topoisomerase type 1 is also equally important. If you generated mice that completely lacked type 1 topoisomerase, it's what we would refer to as embryonically lethal. Mammals cannot survive without type 1 topoisomerase. And the same is true for type 2. So both are important for DNA replication. But here in this case, adjacent to the replication fork, type 2 topoisomerase or gyrase helps to relieve the supercoiling. And our primase will create a short primer. That's the initiating step for DNA polymerase 3. It'll extend the strand and create the complementary DNA. It's important for us to remove these RNA primers because we don't want RNA in our DNA. So DNA polymerase 1 will remove and replace that RNA primer with DNA, and ligases will help repair the breaks in the DNA with new phosphodiester bonds between the fragments. As you can imagine, we talked about how it's really important. We're talking about DNA replication, not in the context of disease, but in the context of chemical and biological warfare, because if there are diseases where DNA replication goes wrong, that cannot sustain life. And so it's really important that any issues that emerge as a response to this have a mechanism to compensate for that. And there's actually one issue that emerges in response to this. As you can imagine, our DNA is linear. It's wrapped tightly in these to form chromosomes. But what's going to end up happening is our chromosomes and our DNA is linear. And so what's going to end up happening is there's going to be a point at the very end of our DNA where we'll have an RNA primer. And what's going to end up happening is that there will not be space for DNA polymerase 3 to have an RNA primer upstream from that, further to the right, to help remove that primer, or to help create new DNA as a result of that. And so what's going to end up happening is these ends of our chromosomes, we call these our telomeres. And so what's going to end up happening is that these sites of our DNA, these ends of our chromosomes, are non-coding regions that have these repeats that are not encoded for anything, but rather sites that are allowed to not be present on further replications of our DNA. So what's going to end up happening is, where this RNA primer is at the end of our DNA, there's not going to be space. It's a space limited issue where there's not enough space in the absence of this primer for there to be an upstream primer for more polymerization to occur. So what's going to end up happening is during replication, there's going to be that overhang missing in the subsequent daughter strand of DNA. And on the opposite side now, there's going to be a space limited problem where the RNA primer is laid down, and there's not going to be space for a subsequent upstream RNA primer to help promote the polymerization of that space. And so that's going to be the other end, the other telomere end of the chromosome, that's going to be missing from a subsequent daughter strand. And these linear sites of these linear ends of our chromosomes that are non-coding, that have these repeats, are called telomeres. This is incredibly important because what's going to end up, what's going to be really important is is in cells where we can't have replication events where the telomeres are removed or shortened. And this is going to be particularly important in stem cells during development, during embryonic development, where in adult cells, our telomeres are continually shortened, and this shortening will lead to a point where our cells can no longer replicate. And so there's an enzyme called telomerase that helps prevent the shortening of our telomeres. And what's really unique about telomerase is that it's an RNA-dependent DNA polymerase, but it has its own built-in RNA template. It does not need the RNA primer to be laid down because it has its own built-in primer provided. And so in this image to the right here, at the very top, as I mentioned before, the telomeres are the ends of our chromosomes, the ends of our DNA, linear DNA, that have these repeats, and you can appreciate that in the bottom figure, that there are these GT-rich repeats that are kind of a protection against this this problem. We call this problem the end replication problem, that we can't replicate the ends well of these telomeres in the subsequent proliferated cells. And so what telomerase is able to do is it's able to prevent the shortening of those strands, because with each subsequent replication, the telomeres are cut off and shortened. So what telomerase has is it has its own RNA primer within that's built in, and that permits it's an RNA-dependent DNA polymerase with its own template. And so in the subsequent image, what you can see is it's able to promote the elongation, with its own built-in primer. And so again, our telomeres are the ends of our linear DNA, or ends of our chromosome, where we have this space-limited problem where there's not actually enough space to polymerize and cover the entire region of our chromosomes. And so it's kind of a built-in safety mechanism where these non-coding regions are shortened with each doubling of a cell. And that's okay, but the issue is we don't want it to get to coding regions where it's going to chop off important segments of our DNA with subsequent doublings. So what ends up happening is that these cells, as the telomeres shorten to a point where telomeres are no longer present, these cells will undergo what we call senescence. For lack of a better phrase, it's basically old age for cells where they can't replicate any further. They're still metabolically active. If they're cells that are adherent to a dish, they're still adherent, they're still metabolically active. They're just not replicating any further. And so for those of you who've worked in a lab so far, and if you're growing out cells and your cells aren't growing any longer, at least I can remember thinking this when I was second year working in a lab, that you did something wrong. The primary cells, they're just not doubling anymore because they become senescent. And so what some people have done to leverage this is people have created what we call immortalized cells, where there's an engineered expression of telomerase that continues to prolong the telomeres and help create this ability to constantly replicate, constantly proliferate and grow. And so, as I've said before, our younger cells have longer telomeres. During development and in endogenous stem cells, they have more active telomerase. But as cells begin to age, they lose telomerase activity. Our telomeres will grow shorter, and cells will lose the ability to continue to proliferate and grow, and they'll become senescent. Are there any questions there about telomeres, telomerase? Yes.
What are some of the issues that could happen with that, like unrestricted telomerase that you're talking about, like taking full cell?
A consequence of higher telomerase activity that cancer cells. Cancer is a situation in which they have much higher telomerase. And I think I'll mention this in a future slide, but I'm happy to say it now. Typically, cancer cells have much higher telomerase activity. As a result of that, will proliferate and grow at much higher capacity. And so chemotherapeutic drugs oftentimes will target cells that have, will target aspects of our replication machinery that are much more active to a degree than our normal cells. And so there's kind of like this window of, you know, above this threshold is really high artificial degrees or not normal pathological degrees of doubling and proliferation, which is where cancer cells will be. But also what ends up happening is like that therapeutic window is also where hair cells will grow to that to some degree that is oftentimes targeted. And that's why people who are treated with chemotherapy oftentimes lose their hair, because they're within or growing that cancer cells will also do. Yes.
This is definitely a dumb question.
Then I'm going to love the question.
Okay. Is this what causes aging?
Yeah.
Actually.
Well, sorry, let me, there are many features of aging, but this is a really big feature associated with aging. And not only that, but a lot of diseases now are being more and more implicated. The pathogenesis of these diseases are being implicated more and more with cell senescence. And so that is one way in which you can easily map telomere shortening with senescence to disease X, Y, or Z. Because, for example, I'm trying to think of a good example here. Well, in the disease eyesight and lung fibrosis or lung scarring, there seems to be some implication that our T cells are not doing what they're supposed to be doing. That's because it seems like some of them are becoming senescent. And this is also an age-related issue. And it seems that these cells, by a product of aging and not repopulating new T cells, you know, it seems that they're tending towards a dysfunction that leads to disease. Yes.
So you're talking about cells that people are like genetically engineering to be like basically immortal. If you like the body could create immortal cells, would we just not age, or would we just like age slower?
Sure. And so that's one idea that people have is like engineering, trying to figure out a way to engineer telomerase to reduce the effects of aging. So like right now, there's like a whole new arm of research. Like the way research has been working is we have academia, we have biotech industry, and now there's like this new third arm of research that a bunch of folks out on the West Coast are creating, where there's a ton of money going into these organizations. A great example of one is, I think it's called Arcadia. It's being launched by a bunch of people who are great, as well as they have 6 Nobel laureates on the board of directors, and all they're doing is addressing aging. And so a lot of them, or a fair number of them, are interested in trying to tackle the question of telomerase expression from different angles or reduce telomere shortening. And so it's just coming at it from different angles, but the central question of aging is what they want to address, and a lot of them are doing it from this perspective. Yeah, I hope that wasn't overload for the, I thought that was a good question. I didn't want to overload you. Yes, yes. Yes, that's a great question. I should have bridged it back to what we've been talking about. So you're absolutely right. It's actually elongating the telomere to permit more, to address this space limited issue that I was talking about, where now polymerization, there's space for polymerization to occur. Yes, that was a great question. I appreciate you asking that. All right, let's press on. So going back to our chemical and biological warfare, the pathogenesis of anthrax. So The most deadly form of anthrax infection that we talked about was the inhalation of anthrax, the inhalation of these spores. And so when it's inhaled, the macrophages within the lung will phagocytose those spores. What's going to end up happening is macrophages are incredibly motile, particularly in the lung. They'll move across the alveoli, move up and across different spaces of the lung. And so they're going to be subsequently transporting the anthrax spores with them. And in addition to that, the spores have, the anthrax is in this spore conformation that protects it and that enables it to wait until it's in a favorable condition to grow. It is now in a favorable condition to grow. Bacteria, as you can imagine from different, I'm sure all of us have had different infections in different ways. Our bodies are great places for bacteria to grow. And so what's going to end up happening is they're going to germinate and proliferate rapidly within the macrophage, and they're basically going to lyse the macrophage. And so this helps, this pathogenesis helps describe how there's robust cell death in response to inhalation of anthrax spores. The ways in which to treat them. So this is going to we're going to try and take a moment to provide some terms and definitions that both help us in this conversation and will likely help some of the pre-med people in this room. So antimicrobials is a broad general term that is that constitutes or includes natural or synthetic substances that are meant to treat some sort of pathogen. Antibiotics are a subcategory. They are a class of drugs that fight against other organisms and are synthesized by other microorganisms. So they're a natural, for lack of a better phrase, they're a natural antimicrobial. Some of the desirable properties of antimicrobials, as you can imagine, we want it to be selective. We want it to promote toxicity selectively, attacking the pathogen or attacking the microbe and not the host. As is the case with almost any drug or any antimicrobial, we'd want it to be stable. We want it to be cheap and easy to administer. Unfortunately, that's not always possible, but it's the desirable property for any antimicrobial you want. This is really important, and we're going to come back to this. You want it to be bactericidal and not bacteriostatic. Does anyone have an idea for what that means, being bactericidal as opposed to bacteriostatic? Yeah. Yeah, that's exactly right. Bactericidal, it kills the bacteria as opposed to stopping or halting their growth. They're becoming static. And so, for example, if you had a tube where you were trying to test the effectiveness of a new antimicrobial, and if in the tube you had 1,000 spores, again, I don't think I provided this spatial reference. Spores are oftentimes on the order of 1 micron in diameter. So cells are typically 10 microns in diameter, 20 or 30. And so spores are typically much smaller than our own cells. And so if you had a tube or a dish with 1,000 spores in it, and if you were testing the effectiveness of a new antimicrobial in a growth condition, if you came back and there were 1,000 microbes in it, that would be bacteriostatic. You don't want that because, typically if you let them grow, you'll see 1,000,000 spores. And it might seem like 1,000,000 spores compared to 1,000, like that's great. It did a great job stopping the growth of them, but you don't want to just stop their growth. You need to kill them in case they try and subsequently become resistant. And that resistance is going to create a lot of issues that we're going to talk about in a moment. But I want to try and finish this desirable properties slide before I jump ahead of myself. And so going back to what I just said, you do not want your microbe to become easily resistant as a result of a potential bacteriostatic side effect of your drug. You want there to be appropriate tissue distribution. If the microbe that you're targeting will oftentimes infect the gut, it's something that you can take orally. But if this is something that affects if If it's something that, if there's the microbe is in the urinary tract, you oftentimes don't want to, you might not want to take it orally. You'll want to try and figure out different ways to provide specific tissue distribution depending on where that microbe is. You don't want adverse reactions, and oftentimes with antimicrobials, allergies are a really important side effect to keep in mind. And lastly, you want there to be an appropriate spectrum of activity. And we're going to take a moment to talk a little bit about what that means some more. There are many of you might have heard about broad spectrum antibiotics versus narrow spectrum antibiotics. Broad spectrum antibiotics typically are typically described as attacking a broad or wide scope of microbes or pathogens, but the narrow spectrum typically are focused on a specific class or a specific group of microbes. And so, for example, one way in which we can see one way in which narrow spectrum antibiotics are defined is this a gram-negative or a gram-positive bacteria, and that will indicate or determine what class of antimicrobials or antibiotics you typically might take. Using broad spectrum antibiotics can be really problematic because what can end up happening is when treating with a broad spectrum antibiotics, what can oftentimes happen is that there can be a subsequent secondary invasion of a bacteria or some sort of other pathogen the secondary infection is resistant to the drug you're using and can lead to what we call superinfection, where there is an initial infection, broad spectrum antibiotics are used to treat it. There's a secondary infection that comes in this sterile environment that is now resistant to this broad spectrum antibiotic you used and can create a lot of problems. And so an example of this, it's not a perfect example, but it's one that can kind of help cast a picture of what this means. In an ecosystem, if you go in and kill all the predators, a series of other secondary issues can emerge where now the predators that control the population of some of the downstream mammals are now eating and consuming all of the grass or livestock in that area. And so like there are other secondary results that happen as a merge of it. That's not a perfect analogy, but it kind of helps bring the picture, helps paint a picture of secondary effects that happen as a result of a broad approach. So there's a lot of words on this page. Oh, yes, Marina.
I just had a question.
Mm-hmm.
Superinfection. It's sort of like, the broad spectrum antibiotics just killed off it might have killed off the bacteria causing infection, but now there's like room for like new bacteria, since they no longer have to compete with this kind of
Yeah.
That's what I was on.
Yeah, and so there's now an environment that they no longer have to compete for resources, basically, for growth, and they can occupy that space now in a way where only those that are resistant to this broad drug can now survive. And in addition to that, these broad spectrum antibiotics can also be bactericidal to our own commensal bacteria that are really important for our own health, our own microbiome. This is oftentimes, it oftentimes might sound like a pop science fact, but it is in fact true that we are, on a per cell basis, we have more microbes than of our own cells. And again, this might sound weird. We have pounds of bacteria on us. I know, it's really disappointing, I'm sorry. But these broad spectrum antibiotics, another consequence of them is that they can disrupt our own commensal microbiome, getting rid of our own bacteria that, again, provides space now for other bacteria to come in as well. And so that's not necessarily a part of what constitutes a superinfection, but rather another consequence of broad spectrum antibiotics. So antimicrobials can affect bacteria at multiple levels. And oftentimes it's desirable when they can attack certain aspects of bacteria biology that don't overlap with our own. So for example, cell wall inhibitors are really helpful because we do not have a cell wall. And so that's exactly what penicillin is. It attacks, it prevents a proper formation of the cell wall. There are also cell membrane inhibitors These inhibitors will disrupt the integrity or the function of these membranes in such a way that it's only unique to organisms or properties of their cell membrane that do not overlap with our own. And finally, we haven't talked about protein synthesis, we will soon, but there are inhibitors that can attack protein synthesis, that can attack metabolism, as well as nucleic acid inhibitors. But what's really important is that these processes overlap with our own. And so these broad spectrum antibiotics can be really problematic because they can not only affect the microbes, protein synthesis, metabolism, and nucleic acid synthesis, it can also affect our own. And so it's really important to make sure that when we're finding ways to target these bacteria, that it's not overlapping with our own processes that can be really deleterious to ourselves. So the antibiotic that we're going to be focusing on in the context of anthrax is ciprofloxacin. Cipro is considered to be one of the most powerful antimicrobials available. And based on the number of cases where inhalation anthrax has been noted, it is the only effective treatment for inhalation of anthrax. And so it falls within a class of antimicrobials called fluoroquinolones. And so what's noteworthy and what's really important to know about them is that based on their chemistry and their structure, they can actually inhibit bacterial DNA gyrase, bacterial type 2 topoisomerase. And so based on what we know about replication, based on what we know about gyrase and type 2 topoisomerase, what do we suspect will happen as a result of inhibiting bacterial DNA gyrase? Liam? Exactly right. And so what's going to end up happening is you'll have these overwound regions of the bacteria's genome. And as a result of that, this supercoiling and overwinding. Yes, Ahava.
Sorry, on the slide with
Oh, sorry about that. The antimicrobial class that Cipro is a member of. So fluoroquinolones. And so this class will inhibit, again, bacterial DNA gyrase. And the consequence of that will be the supercoiling and overwinding of the bacterial DNA. And the consequence of this is that it's going to collapse and break. And so this is an incredibly effective way to attack bacterial DNA replication. So mustard gas, did you have a question? Are you sure? Okay. So now pivoting, that was anthrax. Now pivoting over to mustard gas. We talked about how mustard gas is a vesicant. It's a blistering agent, but it's not entirely known how it acts as a blistering agent. It's believed that the alkylating feature of mustard gas can attack our keratinocytes, and that the keratinocytes respond to that chemical modification in such a way where it creates these blisters on the surface of our skin. But what is incredibly important to know about the effect of mustard gas and its lethality is that it has an incredibly profound effect on our DNA by chemically modifying one of our bases, guanine. What ends up happening is, we described before in the first lecture that mustard gas, that the alkyl chains, the chloride ion will be removed and is very reactive with nitrogens. And so what will end up happening is that these alkyl chains will lose their chloride ions, bind to the nitrogens on guanine, and will actually create a cross-link between guanines that it can physically interact with. And what that looks like actually is that it can create intra-strand crosslinks between adjacent guanines that are next to each other on the same strand. It can create inter-strand crosslinks across different strands, crossing guanines together. And that's a particularly problematic one. And it can create a single a single alkylation on a single guanine. And so as you can imagine, if we have intrastrand crosslinks mediated by mustard gas on guanine, what's going to end up happening is as DNA polymerase 3 is trying to promote the synthesis of DNA during elongation, it is going to hit that crosslink that's on the same strand, and it is going to have either a problem continuing with elongation, or have a hard time creating the appropriate base pair that corresponds to what it's trying to see, this cross-linked guanine. In addition to that, what's going to end up happening is with the interstrand crosslink, where it goes across these two DNA strands here, that's going to create what we call a double-stranded break, which is really problematic, and we're going to describe later on why that is. And so what's important to know, what I want you to take away, is that at these interstrand crosslinks, DNA polymerase will hit that crosslink, and it'll create, as you can imagine, there's also a bit of a conformational change as a result of this, and it's going to create a problem for continuing of the elongation for DNA polymerase 3, but also with the appropriate base pairs. This crosslink of the interstrands are going to create an issue for replication and continue going forward and lead to a double-stranded break. And helicase will not be able to unzip or unmelt the DNA strands here. There are actually chemotherapy agents that use mustard gas. As I described before, cancer cells are incredibly proliferative, and if there's a way to attack them and kill them by targeting their replication machinery, that can be incredibly powerful. And so historically, there have been some chemotherapy agents, one called Bendamustine, that is, as the name implies, similar to mustard gas, based on the ability here of these chloride agents to cross-link the guanines, similar to how mustard gas does. And what can end up happening as a result of other forms of naturally occurring base pair damage. So mustard gas is one way of altering and affecting our DNA. There's also other ways that oftentimes happen, whether it's radiation, UV light, and there are other chemical modifications that can also happen. One option is that the base pair itself, the guanine, is under a class of our base pairs called purines. That's adenine and guanine. So if guanine is lost, you all of a sudden have a depurinated sugar in the absence of guanine. And so that is one naturally occurring way that base pair damage can occur. You can also have the loss of the amine group here on cytosine. And so this deamination, so above was depurination, this deamination can actually lead to a change in this nucleotide, a change in this base, where it's now a uracil. And in addition to that, as I mentioned earlier, UV radiation is one way that base damage can occur. And as a result of this, it can actually create dimers between adjacent thymines. And so these are some of the ways that we can have base pair damage. Now there needs to be endogenous ways for this base pair damage to be repaired. And we have these, we have endonucleases that are capable of detecting changes in our base pairs. As you can imagine, if there's base pair damage or there are changes, that will alter the conformation of the DNA helix. Endonucleases are really good at detecting that. And in response, they'll go in, excise the damaged DNA. DNA polymerase in a 5' to 3' direction will come in and repair that damaged DNA, and DNA ligase will fill the gap and repair the backbone. And so the take-home message that I want you all to know here is that some of the ways in which DNA damage can occur was talked about in the previous slide, and the way in which we repair that sometimes can be through the use of an endonuclease that can go in and sense that distortion in the DNA, excise that portion, and now the DNA polymerases that we've talked about can go in. DNA polymerase 3 will come in a 5' to 3' direction, synthesize DNA, and the ligase will fix those gaps. So I'm going to quickly try and go through double-stranded break repair. So I talked about how one of the consequences as a result of the interstrand cross-linking of guanine can be that there's a double-stranded break introduced. And so those are oftentimes that's a much more dangerous consequence. And so we have endogenous ways to repair that. So double-stranded break repair is not caused necessarily by a lightning strike, that's like is depicted here on the slide, but rather can be from different incredibly powerful radiation events. For example, like getting an X-ray can induce some double-stranded breaks. And so there needs to be endogenous ways to fix that when that happens. And so one way is less elegant, called non-homologous end joining. And so what's going to end up happening is that non-homologous end joining will, in the case of a double-stranded break, will to try and just go over this at a higher level, we'll have proteins that come in that sense that double-stranded break and help mediate filling in random nucleotides to help fix that break. And however, the more elegant way that this can happen is homologous recombination. And the way that this is more elegant is because when there's a resection or a double-stranded break in our DNA, there's actually what's called a homology search. Homology search means there's a search for similar DNA regions or strands that have similar base pairs in parts of our genome. And as you can imagine, we have 2 copies of every gene. So this homology search is searching for the other chromosome on which the same DNA sequence exists. And so once that happens, there'll actually be, we're seeing one strand invasion synthesis. It's actually happening on both strands. Both strands of DNA will go in and invade the homologous gene on the other chromosome, use that as the template now. That is the template for now DNA synthesis here. And what's going to end up happening is we're going to have elongation and ligation to anneal them back together. And so the homologous recombination is a really elegant way, and non-homologous end joining is a less elegant way to repair double-stranded breaks. And these processes, I'm not going to be able to go into this, but these processes are leveraged during CRISPR-Cas9 for genome editing, where we have specific template or guide RNA that help target an endonuclease called Cas9. I don't have time to go further into this just because I ran out of time, but if you are further interested in trying to learn more about DNA replication or find an opportunity to do research on it, here are some members of our community that do that. And with that, thank you for your time, and you guys have a good weekend. Sorry.